Hello everybody, and welcome to another video with me, Ro from Scalpel and Stem. I started a series of videos about how to run a tissue culture business, and from that I've gotten a question regarding how you research, because one of my aspects of the tissue culture business is agreeing to take a look at tissue culture anything. Now I want to add a caveat to that because I have looked into tissue culturing a couple of plants for people, and either it's not worth it financially, or it's something that's already being tissue cultured on a mass-produced kind of level, or I've run into legal issues, licensing that would be required that's expensive, updates to the lab with crazy additions to make sure that you're compliant with these licenses. So those are the times that I would decide against actually tissue culturing something. But the way that I actually research to start something I haven't started before is exactly Google. And I'll show you my screen and we'll go through a quick example. So I'm going to start with the specific example of a vanilla orchid. And although the scientific name was already given to me by the person with this question, hey, how you doing? The best place to start, especially I'm just going to start from the beginning, is you need to know the scientific name in order to get the kind of articles and literature that's going to tell you what you need to know. So let's say I didn't know the scientific name of the vanilla orchid. The first search that I would do is that, and here we go. It's Vanilla planifolia. So we're going to go ahead and type in Vanilla planifolia tissue culture. That's exactly what we want. All right, and so we're going to see first plant cell technology. I'm sure this is relatively easy to follow protocol because they do a lot of great videos and literature about propagation. And if you see one from plant cell technology, it's probably going to be relatively easy to understand and follow. But I'm going to go ahead and go down in case something like that didn't come up. You've got the National Institute of Health, a .gov. I definitely would go to a .gov site. Everybody's search is going to be a little bit different because, you know, all of these search sites are learning search sites and adaptive to what your viewing is. So you're going to see that all of mine are going to come up with a .gov. We've got a .org here, a journals.org. That's another one, scientific articles. So this is going to be a good resource. Here we go. It's another .org. I'm not familiar with that one, but I would definitely look into it. And so since we do have one from Plant Cell Technology, we're going to go ahead and see this efficient protocol because I'm pretty sure it probably is quite efficient. So here we are. And in this article, okay, add. Buy their products. PPM is great. So efficient protocol for orchid. And we're gonna, I recommend reading stuff, especially when you're starting out, because it's gonna give you an idea of what you're working with and help to get a handle on the technology. But if you're not interested in reading, you can also skim down to the part that you're gonna need, which is gonna give you, I hope, here we go. This is the exact explant preparation, which is awesome. And then here we go, shoot induction and proliferation. Yeah, this is literally laid out for you in the easiest way possible. This is specifically geared towards people that have a general understanding of tissue culture but want to move into new species. So definitely plant cell technology. It would be great if they have what you're looking for. Let's go down to like Institute of Gov or the National Institute of Health with the .gov. And in here, it's not going to be as laid out and pretty as it was on the Plant Cell Technology website. We're going to be digging through this entire paper. So cell suspension cultures. They're definitely doing tissue culture. So they are definitely, as you see, establishment and characterization of cell suspension cultures. So they should be showing you exactly how to put it in vitro. And then their little introduction. Again, reading is great, but just for this, it even shows you in this one, there is actually a nice resource for this as well. Callus induction, establishment of cell suspension cultures. So this is when you're going to be putting them in. Growth, and then this will give you different formula. What do we have here? Erlenmeyer flasks. This is just how they put it in to grow. I mean, it's one way of doing it, but it gives you an idea of what you can adapt to yours, because they did include your hormones are in here. You've got your BA, cysteine hydrochloride. I've never used that. 20 grams of sugar. I have been finding that for orchids, you definitely want to go with a lower sugar rate than with your philodendron. And so after you look at that, you look down and see they just finished there. So they didn't acclimate. So you wouldn't have an idea of how to acclimate orchids from this, but you do have an idea of how to get them into culture. Let's try and find something that doesn't have like a really easy laid out. So I'm going to go with Anthurium luxurians, because when I started working on Anthurium luxurians, I did not find a very good protocol for this particular plant. As you'll see, the first thing that comes up is a for sale, even with my preferences. So we're going to scroll down and see what we get. More for sale. Tissue culture of Anthurium. Okay, so here's an example. This isn't the luxurians specifically, but Plant Cell Technology does have an article on tissue culture of Anthurium. So down here, I'm sure they have, there we go. Very nifty, laid out, detailed step-by-step instructions on how to work with this particular Anthurium. But it is not the luxurians. So I would probably start with something like this, because they've already laid it out. And then I would see how that works on the specific species that I'm working with. So it looks like there isn't a very specific protocol right now for the luxurians. And I'm not going to lie, I'm still fiddling around with this. So let me go show you mine. So here's an example. Like I said, I have not had a lot of success with luxurians. Now, getting it into culture wasn't that bad. I was able to get it onto my initiation media. It's a lower sugar, it's basically carnivore media or orchid media. That way I don't have to make that many different types of media, but it's low enough hormones of concentrations and whatnot that the plant survives through the process and slowly acclimates to the in vitro conditions. From there, I take the shoots, and this is in a 1BA. I mean, it's okay. Underwhelming. I put it in one with a myoinostal. So this is 2BA in myoinostal. And it not doing great there. I put it in 4BA, and I think that completely like melted it. Like it was not particularly happy about that hormone mix. It might be changing its mind. So we'll have to see. These were done just about a week ago. Wait, where are we? 2 weeks ago. And then this one just had 2BA without the myoinostal. Inositol, sorry. And this one looks worse off, or as bad as the one with inositol. See, not great. They're kind of noodly and weird. What I'd really want to do next is I think I'm going to try it in a media with TDZ and maybe like a 1.5 BA. So that's a quick rundown of how I do research and how I get my fingers into so many different types of plants. I hope that this helped you, and any other further questions you have regarding anything, starting a business, plant related, tissue culture, mushroom related, go ahead and drop me a comment or message me, and don't forget to like and subscribe to my channel. Thanks. Have a good day.